Carol's highly recognized for her extensive contributions to the field of gene regulation and adaptation in bacteria, starting with the characterization of the heat shock response. More recently, her lab has developed remarkable and novel technologies to provide insights at a global level. In recognition of her many accomplishments, Professor Gross is a recipient of the Waksman Award for Lifetime Achievement in Microbiology and the ASM Lifetime Achievement Award in Microbiology. She was elected to the National Academy of Sciences, the American Academy of Microbiology, and to AAAS, where she was elected as a fellow. Importantly, Carol has also worked tirelessly to increase diversity in academia through leadership of faculty diversity initiatives and interacting with training and providing access to students and postdoctoral trainees. For her contributions to increasing diversity in academia, Professor Gross has received many honors, including the Dr. Martin Luther King Award and the ASM Alice C. Evans Award for the Advancement of Women. And Carol, I'm sorry, but I don't know the title of your talk. So without further ado, Carol.
Thank you. There is a blank screen, I know, but there will be something coming. I needed to just start out by saying how I know Evelyn. Unlike the rest of you, I did not grow up in the field of DNA damage, but I met Evelyn through my late husband, Hatch Eccles. And Evelyn and Hatch, similar to Evelyn and Graham, were really soulmates. Hatch worked not only on DNA repair, but also on Lambda, two things, as you've heard, that are close to Evelyn's heart. So I just want to start my talk by talking about Evelyn. And the first thing that I want to do is to share with you what Hatch wrote about Evelyn in his book, Operators and Promoters, The Story of Molecular Biology and Its Creators, which was actually published after his death. So what he said was, among many contributors, and I think you'll recognize in this the themes that you have heard this whole day. Evelyn Witkin stands out because her 40 years of devotion provided the intellectual boldness, enthusiasm, and continuity that kept the field moving. Arthur Kornberg developed and dominated DNA replication. Witkin nurtured DNA repair and mutagenesis, combining original insights with enthusiastic cheerleading for the field as a whole. I term Witkin the fairy godmother of this research area, to her considerable embarrassment. Whenever my life seemed in a state of disarray, by some miracle, Witkin would be on the phone telling me that everything was wonderful and exciting, and that there was all of this great work to be done. Many others, as you've heard today, also benefited from her personal concern and scientific inspiration. I think that it is extraordinary the extent to which you have nurtured this field, Evelyn. So now I just have to say a few words about Evelyn from my own perspective. So to me, Evelyn, you're a Renaissance woman. You just got it all.
Sorry, I lied.
As has been mentioned, and as you know, is also near and dear to my heart, you're a social justice activist. We've already heard about your leadership of the Bates 7, which led you to actually change your career path from NYU to Columbia. And as you say, that's what made your life, doing that. And one thing that you mentioned was how, you know, this was 1940. It was always in a very ladylike manner. And see, Evelyn is one of these people. She doesn't yell and scream. Things just happen when she does them. Okay, then you were at Carnegie. And this is an example of combining career and family. And you knew Vannevar Bush, this was now Cold Spring Harbor, wanted you to stay on, but you had 2 kids and your husband was away with war. What would you need to stay? She would have to work part-time. So at that time, when it was so unusual, Evelyn did what she needed to do both career and family. And finally, when you got into the National Academy, you noticed that whatever this thing is that you get, I'm calling it a diploma, was not gender neutral. It referred only to men, and you convinced them to change the wording to gender neutral. Okay. I don't need to say anything about what a brilliant scientist you are, because we've heard it. But you make bold moves and seize the moment to create a field and catalyze progress, and has been mentioned doing most of this while working alone. You're a crossover research between science and the arts, just like in the old days. So you're not only a scholar of DNA repair, but you're a Robert Browning scholar who elucidated the influence of the 1678 book, Wonders of the Little World, on both Browning and Darwin, became a member of the Browning Society, conducted research for many years. Yeah, how many people can do all of those things? But finally, you're an amazing parent, grandmother, great-grandmother, and friend. And I've had the luck to have been integrated into your family, to, in particular, really know your 2 granddaughters, one of whom worked in my lab for a while, and the other one I've talked to is currently getting her MD degree. I can't really say very much about this because you have to experience Evelyn in these roles to know what it means. So thank you, Evelyn. Now, what I'm going to talk about is not related to DNA repair, but to me, it's I chose to talk about this because I think it is a very crucial area that's just coming into being now. And so the title of my talk, which is not very enlightening, is New Insights into the Cellular Economy of Bacteria. And I'm really going to talk to you about new genetic approaches that are now allowing us to study essential genes in vivo for the first time. So essential genes are clearly very important. They encode the central features of cellular life. They're interaction hubs for cellular networks. They're evolvable depending on the environment and genetic background, and we know that that's a quantitative trait. And importantly, they're a major consumer of cellular resources. The most expensive thing that the cell does is to make protein. That's where it uses most of its ATP. And although essential genes are only 5 to 10% of the genome, they're 50% of its cellular protein synthesis. And so that makes it important to understand how the levels of expression of these genes is set. But they're hard to study. So understanding the fitness landscape under which expression levels of essential genes are optimized awaited the development of a method to reliably titrate gene expression. Can't just knock them out. It's not very elucidating if cells are dead. Sorry. What did I do? Okay, so the advent of CRISPR interference allowed us to focus on the essential gene set, which I'm calling the essentialome. And let me just briefly describe this. system. It's a system with a protein component, which is an endonuclease, Cas9, which in this application has been deactivated by mutation. So it's dead Cas9. And the other aspect of this complex is an RNA that binds to Cas9. And then there's a bipartite recognition. First of all, the Cas9 protein itself recognizes a 3-base sequence, which is called the PAM. And if that PAM happens to be adjacent, the DNA that's that can base pair with this orange part of the RNA, you get a DNA RNA hybrid, and the combination interferes with transcription. Okay, so how can we go from that to getting graded knockdowns? So basically, by using these guide RNAs that have single mismatches in their base pairing region, we created an allelic series of guide RNAs that target essential genes and allow us to systematically titrate their expression. And then this allowed us to examine essential gene expression fitness relationships. So here are the players in my lab. John and Melanie were graduate students. Young Mo is a long-term researcher who did a number of experiments in the paper. Horia Todor is a postdoctoral fellow who really spearheaded the project in this direction, a very creative person. This was done in collaboration with Marco Yost, a former postdoc, jointly between me and Jonathan, who's now a faculty member in HMS micro department, and my long-term collaborator, Jonathan Waksman. Okay, so here's the outline. First, of course, we had to develop the method. And in order to do that, we made a comprehensive series of single mismatch guide RNAs against GFP. And then we could directly measure how much each guide RNA knocked down GFP activity so we could figure out its efficacy. And we did that both in B. subtilis and E. coli. And so our first question was, is knockdown consistent across bacteria? And it is. So basically, this CRISPR system works the same in, if a mismatch guide has a certain effect in E. coli, it has a similar effect in subtilis. And then the next thing we needed to do was to ask whether we could use this data to develop a predictive model of the guide activity. And the answer to that was also yes. And it was a fairly simple model. Okay, so now we have a way of making guide RNAs that will titrate essential genes. So here's the experiment. First, we designed systematically compromised guides to titrate expression in B. subtilis and E. coli. Just 1 second here. Oh. And then we did pooled fitness experiments to determine expression fitness relationships. Okay, so here's the design of the experiment. So what I'm showing you here is the essentialome, these approximately 300 genes, both in E. coli and in B. subtilis. And for each gene, we identify the PAMs, that's where the Cas9 can bind. And we made guides, 9 single mismatch guides for each of the first 10 PAMs. So we had 100 guides per gene that could knock it down to various extents. And so in theory, for each gene then, depending on the efficacy of the guide, you only get in these mismatch guides, only get a little bit of knockdown, and with no mismatches, you get full knockdown. So now we have these 30,000 pooled guides for E. coli and B. subtilis. We put them into both E. coli and B. subtilis. And then we have separate flasks, either with E. coli or with B. subtilis. And at that time, we start the knockdown by inducing expression of dCas9. And then we look at the distribution of guides across bacteria, because each of these guides really barcodes the bacteria. At the beginning of the experiment, before knockdown was initiated, and 10 doublings after exponential growth. So what you can see is this is sort of classic genetics, but with a mechanism to make it genome wide to answer interesting questions. Okay, so now imagine this orange one. It has a certain distribution at the beginning, and suppose it didn't grow. So it's going to be present much less at the end. And likewise, if it's sick, it'll be decreased less. If it's mildly defective, not that much, and wild type doesn't change. Now we can quantify the growth defect by looking at the relative fitness, which is the average fraction of wild type growth. So a relative fitness of 1, same growth as wild type. 0.5, it's half the growth. 0, the cells aren't growing at all, and less than 0, the cells are dying. So then we made these expression fitness curves for every essential gene in B. subtilis and E. coli. And our first finding was that the expression fitness curves co-vary by pathway. You can see that, for example, if you knock down cofactor synthesis, so if even when you have a very high predicted, let me just say that the axes on these are all relative fitness on the y-axis. So 1 here is unimpeded growth, and on the x-axis it's predicted activity. So if the predicted activity is 1, it's full knockdown. And you can see that fitness goes down only slowly if you're knocking out a cofactor. It goes down more rapidly if you're knocking out something in translation. And if you're knocking out peptidoglycan precursor synthesis, it seems to be almost bimodal. Either the cells are alive or they're dead. But when we looked at E. coli, we saw that E. coli basically had the same pattern. So these patterns of expression are conserved across 2 billion years of evolution, either by convergent evolution or by conservation. Okay, now let's look and see what, let's think about what these curves mean. So the fact that fitness goes down only slowly as you're decreasing expression is actually not that surprising. It's not surprising that both species are robust to knockdown of essential cofactor genes. It's actually consistent with what happens when you try and make deletions of these genes. Most of them will regrow as will form small colonies, but then they won't regrow. And if you look at these curves, you'll see that there are a few that seem to go down faster in both B. subtilis and E. coli. And all of those genes have roles in addition to being cofactors, so they're decreased more rapidly. And so this suggests that these enzymes and or their products are greatly in excess of what is required for viability, and it takes a long time for them to be diluted out by growth enough so that the cells die. What about translation? Well, both species are linearly affected by knockdown of translation genes, suggesting that the logic of regulating ribosomal protein synthesis is conserved. And I should say that what we imagine is that when you're knocking down components of the ribosome, you're actually limiting the number of ribosomes. But in amongst these genes are also genes that, let's say, knockdown factors that are involved in translation, but not the ribosome itself. And that's consistent with work from Terry Hua's lab showing that that you are probably eliminating, decreasing the rate of elongation. So limiting translation in that way. Okay. What about this peptidoglycan precursor synthesis? This was actually very surprising to us because we know that a gram-positive cell wall has a very thick peptidoglycan layer and no outer membrane, and the gram-negative cell wall has a very thin peptidoglycan layer and an outer membrane, and that actually is the basis of the gram stain. So it was a little surprising that they behaved the same. And for one of the genes, the MUR-AA or MUR-A gene involved in peptidoglycan precursor synthesis, we actually did a comprehensive knockdown analysis of every possible PAM in the gene. So thousands of different representatives. And I think this shows you very clearly that you go from either 100% fitness to almost no fitness or death. What's known from a lot of work is that peptidoglycan enzyme synthesis is high and it's constitutive, it's not regulated. And so it suggests that cells can't regulate the flux through the pathway. They keep on growing fast until they lyse. And why is that? That suggests that flux through this pathway may be used to set another cellular parameter. And indeed, there's recent work that suggests that flux through this pathway is used to set surface to volume ratio and also adjust metabolism to growth rate. So that may be why the cells tolerate dying in order to sense this signal. Okay, and then we then asked whether there are any differences between E. coli and B. subtilis. And there were 9 genes that were significantly different, and 7 of them were in other aspects of the peptidoglycan synthesis pathway. So far I've been telling you about peptidoglycan precursor synthesis, and those are the yellow genes in this pathway, and that's what I've been talking about, number 3. But what you can see is that genes that feed into making the precursor of the building block, the UDP glucanac, genes that are involved in this modification of DAP, and the genes that take the precursor and incorporate them into the peptidoglycan are all much less sensitive to knockdown in E. coli than in B. subtilis. And why might that be? Well, we don't have a complete answer yet, but there are several possibilities. One is that there's feedback of synthesis in E. coli that adjusts, even though we're knocking things down, the cell is compensating by upregulating transcription. And that seems to be the case for the GLIM operon. And it's also the case, we and others have demonstrated for MREB. So that's part of it. A second consideration is that gram-negative bacteria recycle peptidoglycan during exponential phase, but gram-positive bacteria don't. And we have evidence that disturbing the flux of recycling does alter sensitivity. It makes E. coli more sensitive to knockdown. And finally, E. coli and other gram-negative bacteria have an outer membrane, and we now know that that outer membrane is partly load-bearing in addition to the peptidoglycan layer, and that may have an influence as well. So what I've told you today is we've developed a predictive model for mismatched guide RNA that's universal across bacteria. expression fitness relationships of essential genes vary from linear to bimodal, but are generally conserved by pathway. Deeply conserved homeostatic constraints underlie the expression levels of essential genes. And just parenthetically, using this technology facilitates drug discovery and lead optimization by identifying cellular vulnerabilities and allow you to do a lot of experiments in a single test tube. And I think this is only the beginning of our studies in this area. There's clearly much more to do in terms of asking how different environments and different genetic variations in cells affect the expression fitness relationships, and in terms of going to additional organisms to carry this out across bacteria. So thank you very much.